Hello everyone, I'm back. This is another Science Talk episode, and this is Nicole. I hope everyone is doing well and actually feeling awesome because it's almost the end of the week. It's Thursday and Friday will soon be here. I don't want to make this video too long. I just wanted to highlight another Science Talk. This is a research article that one of my good friends, Lisa, published a while back, 2016. This was published, as you can see, December 12th online, December 12th, 2016, when it was accepted as a manuscript. And it's from the journal Molecular and Cellular Biology by the American Society for Microbiology. And the title of the article is extremely long, of course, but there's a lot of information, good information in this article, and I'll get to the actual data in a minute. The title is Targeting the Nuclear Cathepsin L CCAAT Displacement Protein / Cut Homeobox Transcription Factor Epithelial Mesenchymal Transition Pathway in Prostate and Breast Cancer Cells with the Z-FY-CHO inhibitor. And so it gives the author's names there. And this research was conducted at Clark Atlanta University in Atlanta, Georgia. There's the PI's name for correspondence. And I'll leave this link in the description box. Now that I'm thinking about it, let me go ahead and copy that so I have it for later to put in the description box. So here's the abstract. In the abstract, I also want to use this video as a way for people interested in science and research, specifically cancer research, which is an area I'm interested in, and I work in, to see how beneficial it is to read research articles and the ways you can go about reading these articles pretty quickly. So the abstract is a summary of a summary of the significance of the research, why it was done. So the purpose of it, you recognize an issue, a problem, some type of genetic or cellular phenomenon, and then you conduct research with a hypothesis where you state a claim and then you conduct scientific experiments, whether in vitro, meaning in a petri dish, or through cellular culture, or in vivo, meaning that it's on mice or humans, to do your analysis, and then you give your conclusions from your data, whether it supports your hypothesis or not. So that abstract states the epithelial mesenchymal transition, or EMT, promotes tumor migration and invasion by downregulating epithelial markers such as E-cadherin and upregulating mesenchymal markers such as vimentin. Cathepsin L, or CAT-L, is a cysteine protease that can proteolytically activate CCAAT displacement protein cut homeobox transcription factor, or known as CUX1. We hypothesize that nuclear cathepsin L may promote EMT via CUX1, and that this could be antagonized with CAT-L, or cathepsin L specific inhibitor, Z-FY-CHO, and that's a chemical composition name. Mesenchymal prostate, RCAP-M, RCAP-E overexpressing snail and breast cancer cells, MDA MB468, MDA MB231, and MCF7 overexpressing snail cancer cells express lower E-cadherin, higher snail, vimentin, cathepsin L activity, and P110/P90 active CUX1 form, as compared to epithelial prostate, RCAP-E and RCAP-Neo. And RCAP-Neo is the control vector. And breast cancer cells, MCF7 and MCF7-Neo, and that Neo is short for neomycin, which is the control vector. Again, these cancer cells. There was increased binding of CUX1 to snail and E-cadherin promoter and mesenchymal cells as compared to epithelial, prostate, and breast cells. Treatment of mesenchymal cells with cathepsin L inhibitor, Z-FY-CHO, led to nuclear and cytoplasmic relocalization of cathepsin L, decreased binding of CUX1 to snail and E-cadherin promoter, reversed EMT, and decreased cell migration invasion. Overall, our data suggests that a positive feedback loop between snail nuclear cathepsin L-CUX1 drives EMT, which can be antagonized by Z-FY-CHO. Therefore, Z-FY-CHO may be an important therapeutic tool to antagonize EMT and cancer progression. So that's the whole abstract. And then they also give you on their website other articles that they recommend you read because they are related to this particular article. And I always look at those at the bottom because they're usually very interesting. Some of them are in Journal of Clinical Pathology, Molecular Cell Biology, Journal of Virology, the Gut, etc. So let's go to the actual download. Okay, so here's the download. It shows the when it was accepted, when it was posted online, gives a copyright 2016, and here's the figures. So the first figure, I'm not going to go through all of them because as you can see, let's scroll down. There's the second figure, there's the third figure, the fourth figure, fifth figure. I know they may all seem the same, but they're not. Here's figure 6, figure 7, figure 8, and here's the last figure. So I'm just going to go through the first figure and the last figure, and you can go through all of them at your leisure. You can download the PDF. There was a section on the website. Let me go back to it. Here at the top of the website, the top of the page, let me minimize it some. So here on the right, when you come on the page, you'll see it'll look like this. It'll look like this. And you go over here to this section, just to the right, where it says PDF. So you'll click PDF and it'll download for you. It'll show you similar articles. It'll show you articles from that author. It'll take you to their ResearchGate pages, things like that. So let's go on to the first figure. I'm going to zoom in. Figure 1A. So what they did was a panel of, let me make it even bigger if I can. All right. What they did was a panel of 1, 2, 3, 4, 5, 6, 7, 8, 9 different cells that they have here. Here we go. RCAP-E, RCAP-M, MCF7, MDA231, 468, RCAP-Neo, RCAP-Snail, MCF7-Neo, MCF7-Snail. And they show the protein expression of various EMT markers. E-cadherin, snail, vimentin, pro cathepsin L, pro cathepsin L, mature cathepsin L. Then they give CUX1 and all its different isoforms, and they use the alpha tubulin at the bottom as their loading control. So what do I mean by this? EMT is a process that tumor cells go through. There are 3 different ways that, I shouldn't say tumor cells only, 3 different ways that you have EMT. One is with tissue development, which is normal development, embryonic development, when you're in the embryo, in utero. The second way of EMT is through wound healing, where you have a wound, you have a cut in your skin, and the cells have to come back together to close the wound. You don't see that happening. It happens usually under a band-aid or a gauze, but then a few days later you take the gauze or the band-aid off and your wound is healed. Well, those cells had to change genetically in order for the wound to heal. And the third most studied way that you have EMT is through tumor development and progression. The same genes that turn on during embryogenesis and during wound healing also turn on during tumor development and progression. When tumors grow and they begin to expand outside of the original organ, such as the prostate or the breast, certain genes are turned on, certain genes are activated and deactivated, and that's what causes that tumor progression. So some genes like E-cadherin and snail work at the transcription level to regulate the mRNA and therefore protein that is translated to have the function change in these cells to become more worse tumor cells in the human body. Other genes such as cathepsin L literally work as enzymes to cut a substrate and help to release the cells from their environment. And as they are released from their surrounding environment, they move away from the primary site, such as the prostate or the breast, and they're able to metastasize. They're more likely to metastasize into different parts of the body. Metastasize just means that they're able to move around, move freely through the bloodstream or the lymphatic system. So what they found here is that some of the genes are expressed at higher levels in some cells. And so in B, they looked at the activation of these genes, of the genes that actually cut themselves free from the surrounding environment. Cathepsin S and cathepsin L is expressed at different levels in the cells, more likely to be expressed in cells that have higher SNAIL, like MCF7 SNAIL, RCAP SNAIL, and the triple negative breast cancer cells, MDA MB231 and MDA MB468. Now, why am I doing an article about breast and prostate cancer? Well, as we know, as you should know, breast and prostate cancer, the more aggressive forms of these cancers, kill and are diagnosed in African Americans at a higher rate than any other racial group in the world, especially in this country. So it's important for us as African Americans to do the analysis necessary to produce new therapies and understand previously studied concepts to help develop better therapies in going into the future. So we need to understand how these genes are working. This, indeed, it shows the differences in expression in the cytoplasm versus the nucleus. So if you look up, Google the organelles of a cell. Organelle is spelled ORGANELLE, organelle. It's the word organ with ELLE at the end. You see that, for instance, CUX1, all forms of it, the P200, P110, and P90, are all expressed higher in the nucleus, which is the brain of the cell, compared to the cytoplasm, which is all the other parts of the cell. So this is an extremely important gene to compare with SNAIL, for instance, because SNAIL is a transcription factor that only functions in the nucleus, the brain of the cell. So there may be a connection between SNAIL and CUX1. So that's what they wanted to look at next. Now here in the last figure, last part of this figure, excuse me, I'm trying to make it a little bigger for you to see it. Well, I'm gonna look that part there. Let's go right here. Okay, they used RCAP-E, RCAP-M, RCAP-NEO, and RCAP-SNAIL, and they look at the expression of E-cadherin, SNAIL, and vimentin. So E-cadherin is a gene that would be expressed in cells that are less, tumors that are less aggressive, and SNAIL and vimentin are expressed in cells that are more aggressive, tumors that are more likely to migrate and invade surrounding tissues and metastasize. And what they found is if you overexpress SNAIL, that transcription factor that we talked about, in these cells, you don't have expression of it. So the red is the expression of E-cadherin. And as the cells become more aggressive, you lose expression. The last box, it's blank. And this is immunocytochemistry. This is immunofluorescence that they're using. And you can look up what a immunofluorescence is later. If you read through the whole article, you'll see how they conducted these experiments. They have a method section. The next gene they looked at was SNAIL. And as you expect, these cells, they are cancer cells, but the ones at the end are more cancerous because they have expression in the nucleus and the cytoplasm. And the RCAP-M, M stands for mesenchymal, and E stands for epithelial. So the more mesenchymal you are, the more tumorigenic you are, the worse the tumors are for patients. So if RCAP-M and RCAP-SNAIL were in patients, these patients would be very, very bad off. They would require surgery. They would require treatment. And lastly, you see the same expression with Vimentin, similar to SNAIL. SNAIL is in green and Vimentin is here in red. Okay, so you see the same expression pattern where it's high when you have SNAIL or when the cells are mesenchymal. The last one here, excuse me, I forgot about cathepsin-L. Cathepsin-L is the most important gene for Lisa in her studies that she's doing even now. She just received a grant, I think 2016, 17. So this is the last thing, and she shows that across the board you see high expression levels of cathepsin-L. So regardless of whether the cells are epithelial or mesenchymal, there's high expression in the nucleus and the cytoplasm. So that was a target for the rest of her studies in this paper. And all of these studies go on to use the treatment, and you see the treatment reducing expression at the protein level on the left for A and B, and when you look at it under the microscope through immunofluorescence as well. And then all of these are supplemental to the previous two figures. And the last figure, let me come on down. The last figure is summing up what they did in the study, what it means. So the SNAIL transcription factor they don't know, that's why the question mark is there, may activate nuclear cathepsin-L, but this can be blocked using the Z-FY-CHO drug, and that's the chemical structure of the drug. It has 3 aromatic rings on it. Those hexagons are aromatic rings, and it is able to block nuclear cathepsin-L activity. If you don't block nuclear cathepsin-L activity, nuclear cathepsin-L will activate P21 isoform of CUX1, and that can bind to both P90 or P110, which are transcription regulators that then come with their the little legs they have there. They bind. They use those little parts of the protein to bind to SNAIL transcription factor promoter. And a promoter is just a nice word for part of the gene that helps activate making more of that gene, right? So now when you activate SNAIL, SNAIL makes protein. It makes more of itself. It's self-promoting and it creates itself through DNA replication and through mRNA transcription. Then those two parts that activated SNAIL can then bind with SNAIL onto SNAIL's target, E-cadherin. And that red X means that it's blocking transcription. So now E-cadherin can't be made. So E-cadherin goes down. And I think in one of the figures they showed up at the top, I think they showed E-cadherin going down when you use this drug. So this all leads to increased epithelial mesenchymal transition, and you have higher migration and invasion of tumor cells. So when you have this EMT and higher migration and invasion of tumor cells, this is what the doctors try to avoid, because this is what causes them to have to diagnose you at a certain stage of cancer and then treat you with various drugs. And this paper has shown that using the Z-FY-CHO drug to target this one gene, cathepsin-L, you can reduce the level of migration and invasion of these tumor cells. And this is the whole paper. Here's your abstract that I read earlier. The introduction is it's very short. If you can see, it's very short. It's not long. So you can go through this article. This is their actual manuscript that was submitted. And all the numbers on the side are the line numbers, of course. They go through their methods, how they did the experiment. That's important to look at. How much of certain drugs they used, their statistical analysis. Then they actually give you the written form of the results. So the results section here that's written is summing up what you saw in the pictures. And the results, the data is very clear. The figures are clear. That's why I could go through them so easily. And then they give their discussion section, which sums up everything in the paper and gives references to other papers relative to this study. And finally, at the bottom, you have your references. And then you have, your figure legends as well. So that's the end of the paper. Let's go back up to the top. So if I was starting to read an article, the first thing I would do would be to read the abstract. I would read the abstract first, and then go and read the figures, go through the figures and see if I can understand what it is they are trying to say in their figures, and go from there. So let me know what you think in the comment section. Like, comment, subscribe, share the video. I hope that you enjoyed this. If it was boring, please let me know. But this is a very interesting and important topic to discuss. Research that's being done by African American scientists in the United States who don't get a lot of recognition, but we are out here and we are doing the research, specifically research that affects our community. And with that, I'll leave you with goodbye. Have a good day, and I hope to see you all soon. Bye.